trouble, but thinks he can hear the mic. Say hi.
Oh, the what's his face guy?
Yeah,
Why is this so bad?
yeah. Lee John.
Alright, so the headphones are currently on. Should be all set.
To do what?
You're on.
Hello, good afternoon. So, good afternoon. Can you guys hear me?
Yeah, I can hear you.
Great. How are you?
Good, how are you?
Doing well. Good afternoon, everyone. Did you hear me? Okay. We are out here at Edgewater. We just took a swim. We went 3 miles out. It was just a typical morning swim for us. Dried off a little bit, got changed. The water's a little brisk.
Just went to the Cleveland water intake, right? Just still out there.
Yeah. It's actually a nice day at the beach. It's not too breezy. The sun was poking out a little bit. Miss Danita is running the video for us. So make sure to thank her in the chat. How's the video looking? Too bright? How about it? Does it focus in on us at all? All right, working on the video issues. But in the meantime, Mr. Tout, do you want to start off?
I guess I'll start off with a quick overview of what we're doing. Today, we are looking at ways you can test for and collect data on biodiversity and on population sizes in theory, ways you can extrapolate that data to get population estimates. We're going to focus on a few different techniques because they are ones that you guys could use for your IA. We're going to start off with just simple random sampling and move on to then a stratified random sampling, which is, as opposed to just completely random, you pick a couple of different areas specifically, and then you randomly sample within there. The third major type is sorry, let me make sure I get the systematic sampling. And we're actually we're going to teach you 3 ways, potentially, if we have time. We're going to go over 3 ways you can do systematic sampling. So think about systematic versus random, right? So systematic, there's a line transect and a belt transect, which are very similar. We'll show you slight differences between the two. And then if we have time, we'll get into the very last lab on this assignment, which is using a quadrat sampling, which is divided then within the quadrat. In case you were curious, we do have our first deer sighting. So we have our first example of a macro vertebrate species. We have our first large animal, not the target group we're looking for today, but we are going to talk about how to sample particularly plants and invertebrates, that is bugs. How are we coming with the video, Mr. Dutton? Has everyone opened up your Google assignment? And now I'm trying to remember for sure which number it is.
20.
It is 20? I was going to guess 19.
Yeah.
Thank you. Yeah, open up assignment 20 in Google Classroom. And from there, you should be able to see a Google Doc which has these sampling methods, ecological sampling methods. We have our first canine sample coming. Kidding. So to clarify if you're looking at this first page, right, our introduction is species diversity, right, is a measurement of the number of species and their relative abundance in one or more habitats. So we want to measure both the richness and the evenness, right? We want to try to get information and data on both those things. That is the number of different species, how rich is the biodiversity versus the relative numbers of individuals of each species. So do you end up with, say, a cone-shaped population pyramid? Do you have, one species in excess and another species with just 1 or 2 examples? And then potentially, hopefully, like among your herbivorous bugs, you have maybe some evenness. In terms of the Simpson's Diversity Index, I have good news. You don't need to be able to interpret that formula. We want you to be aware that there is some math going on, some statistics going on, and some calculations going on. But you're not going to have to actually use that calculation in terms of, you're not going to have to plug in and solve these for yourself. We have a spreadsheet that does the math for you, thanks to Mr. Dutton. So what our main goal for today is literally just to collect some initial data in these data tables, starting on the second page under types of sampling. So if we look at this first diagram, simple random sampling versus stratified random sampling. Sorry, is that a question? Or? Okay. Simple random sampling versus stratified random sampling. Simple random is what it sounds like. If you were along a creek, you would literally just pick random sites. You're not going to necessarily walk up and down an entire length, say for instance, if this were our creek, right? I wouldn't necessarily walk a 3-mile stretch and take data along all 3 miles, right? That would be way too time intensive. Rather, you would pick a few specific sites that you would sample. Oh, we're up to 2 deer. Thanks. I don't know. I'm assuming you can't get the 2 deer on the video, right? No? Okay. Oh, right there. Right behind you. They're creeping up behind you. Anyway, so So stratified random sampling means you recognize that there are maybe a couple of different ecosystems along the stream, right? A couple of specific sites that are significantly different. Sorry, the mask is not cooperating. So recognizing in this example, right, there's, say, along the stream, maybe a sandy sections, really muddy sections, and some gravelly sections, and that you specifically try to pick two random spots from each, right? So now you've stratified your random sampling. It's not just simply random, but you're recognizing. Oh, go ahead. Let's go with the random sampling first. Simple random.
Oops. Simple random sampling first, and what we'll do is we'll determine. Mr. Tout has his little softball here, his styrofoam softball, and he is going to determine a couple spots that we're going to sample for biodiversity.
Am I going up the hill? Right.
Yeah.
Okay.
All right. So that's spot number one.
That's a perfect spot, by the way.
It's a perfect spot. And then before Mr. Tout moves into that spot with the, I'm going to also, I've got beanbag here, and I'm also going to find a random spot to sample. The beanbag is red, so it won't blend in, all right? And then what we're going to do now is we are going to use the quadrat. Okay. And the quadrat frame is basically a 3 foot long by 3 foot long frame. And so you can actually do a wide variety of things with this. The first is to just kind of like organize a space for you to.
Do you have pencils?
Yep.
Okay.
You can organize a space for you to actually do your random sampling. I have another way of doing this. And
you got them.
You know, we have quadrat frames that you can borrow to do this kind of sampling. And then something else you can do, if you don't want to make any sort of 3 foot by 3 foot grid like this, is you can take a bunch of pens and pencils. So I'm going to do that for the other sampling site I have over here. We're going to be kind of careful because this is a protected area, so we're not going to try to trample everything that's in here. But what I can do is I can actually mark off the 4 corners of the area that I'm going to sample. And you can use any measurement that you want. We're not particular. If you want to use lengths of your boot, lengths of your shoe, if you want to use pieces of paper to measure, those are all valid measurements because we're having you do labs on your own. So there I stuck 1 pencil in the ground. And then I'm just going to kind of use my boot to measure off about 3 feet, and that's the next corner. I prefer 1 meter. Or 1 meter, which is about 3 feet, right? And so that is about right there. And then once I have 3 corners, then I can figure out where the 4th corner is. I'll zoom out a little bit, so hopefully you can see that. And then the 4th corner would be over here.
And to clarify conceptually, conceptually, what Mr. Dutton is doing is he's just making this quadrat, right, in the same sort of, the same concept of space, right, that we have using the PVC piping, right? It's just, obviously, you likely don't have these, but you might have 4 pencils that you can, you know, use to just outline your space.
So now at this point, what Mr. Todd is going to do is he's actually going to count the number of species inside of that square. And what that's going to do is that's going to give us a count. so that we can start to get the amount of biodiversity in this area.
So we have a lot of tall grass, right? Several bunches of it. So in the example, there's a dandelion listed. I am not an expert on the different species of grass, right? But you don't have to be. You can just start off with sort of identifying or assuming, if I take a closer look at this, right, there's at least one species of grass here. And then the question is are there multiple species, right? And if you can find a difference, you're probably looking at two different species of grass. If you think if they really look very similar, they're likely the same. I am mostly seeing one species of tall grass here. I do have one grasshopper who just left my quadrat, of course. But he counts. But he counts. He was in there. I'm really annoyed that because of this random sampling, let's say this species right here, if you can see these burrs, is outside the quadrat, so I don't get to count him. But I can certainly say that this guy, right, which is actually his base is inside, is quite a different species. Certainly not a tall grassy species, right? This is, well, any thoughts, Mr. Dutton? Some sort of, I guess.
I am no botanist.
Yeah, so I would just say some sort of flowery bush. It's got some small white flowers on it, right? And so again, just a quick description. It does not need to be a perfect identification of, oh, this is the Latin name. No, that's not what we're looking for here. But just having some of the basics. So I do have one more example of this white flowered plant. And then I have, as you look closely, it appears that there is one other species that doesn't have anything flowering on it right now, but the leaf is clearly, the leaf of this sort of weed or grass is significantly wider and shorter than the taller grasses I identified at the first. So at this point, I've spent a couple of minutes trying to identify how many different things are here, and I've definitely identified 4, right?
4 different species. So then the second part of this is identifying how many of each species you have. So that's really easy for like a grasshopper because he only found one grasshopper. And it's really easy for the white flowered plants and also the were those are the same one though, right?
What?
Those 2 white flowered plants?
No, there's 2 separate ones. So we have 2
okay.
white flowered ones. Yeah, 2 separate white flower ones. I think of this wide-leafed weed. I think we really have, I'm going to actually say 3, but they're just much younger and a little bit hidden.
When you're talking about any grasses or anything else that is numerous, like the grasses that are inside of this frame, then it's most important that you estimate down to the nearest like 10 or 100 or 1,000. If you think there's 1,000 in there, then put down about 1,000. It's not important that it's exactly 800 or 1,200. If you think there's 100, it's not important that there's 80 or 120, not for what we're doing. So it's important to be able to make some sort of estimate and not have an exact count. However, if you have any species that you have at least one of, you need to list that.
Yeah. So to put that information into this example, I would say, right, I have 3 different plants that are this sort of wider leafed weed, and then I have at least 8 or 9 sort of bunches of tall grass, and each bunch has at least 20 blades. So I'm just going to say, let's say I have 8 bunches of grass, right, that are pretty evenly distributed. If there's 20 in each one, math is hard, but that's about 160 blades. So I might even say 200. And again, the difference there is really not that important. Just to recognize it's not 3, it's not 1. It's in the range of about, say, 200. Okay?
So then how many different species did you guys count that Mr. Tout identified in this frame? Mackenzie says 4. And then what we're going to do is we're going to write down what our names for those species are and the number we found for each one.
And again, the examples we have in there are dandelion, crabgrass, and clover. And you see numbers next to those in the other column. So in the blank space underneath that example, there's a space for you to record these 4 examples.
And you should be recording that right now because this is forming the basis of the data that we're collecting as a class, which is going to be a huge portion of your lab.
So I can't turn on my camera, can I, since I dialed in? Or is there where can I turn on my camera and show?
You have to have Zoom. It's okay.
Okay.
So I'm going to move over to the one that I marked off.
Do you want the quadrat, or do you want to just keep it as the pencil?
Nope. We're gonna go low tech here, and we're gonna stick with the pencils. And I'm gonna try to not step on the pencils as we go through this. So we've got one pencil right here. Oh, look at that. Hello. Do you see him? There he goes. But he counts. He was in my quadrat. So that's grasshopper number 2, and we're just going to continue adding on to what Mr. Tout had. Is that a decent video? Let me know if it is, and I'll stop. So we're going to continue adding on from with these species from what Mr. Tout had. Now, you notice that now I'm going to take a bigger view here so that you can see some of the different plants we have. This looks very different from what Mr. Tout had. So I'm going to call this a tall, flowery shrub. I'm just going to say tall, flowery shrub. I don't know what it is.
Not a shrub.
Well, it's a shrub now. We'll call it David. How's that? All right. Because again, we just need to keep in mind what different species we have. And so then there's this, it's not goldenrod, I don't think, but it's got these golden buds on top. And so this is another species, tall with golden buds. That's what I'm going to call it, AKA Peter. And we've got in this quadrat, and I'm going to zoom in so that you can see why I'm making the decision I am. So here is one corner of my quadrat, and I see right along here that these 2 guys are inside. So, and they're all the same species. So I've got 1, 2, 3, 4, 5. So it looks like I've got at least 5 of those, the tall goldenroddy kind of things. What else do I have inside here? I'm trying not to kill everything. I have this guy, and kind of looks like a sage. Can't smell through my mask. And I don't know, it looks kind of like brown, ashy down here. So this is my brown, ashy friend. And I see 1, 2, 3, 4, 5, 6 of him. So I'm adding him to the list. And then there's this small little spiky guy. That's pretty cool. And I'm seeing 1, 2, 3, 4, 5, 6, 7, 8. I'm seeing a lot. I'm going to estimate 20 of these small little spiky dudes. And we're going to call the small little spiky dudes
Mark.
Who?
Mark.
Mark? Okay. Mark sounds like a good name for a small little spiky dude. There's more of the grasses. I'm not seeing a whole lot of the grasses in here. And that's outside and there's almost looks like a plantain-y kind of weed. So that wide-bladed weed. And there's probably some more things I'm not seeing. But if I spent more time in here, I could probably see a few more species. This is really species rich. And so by just examining 2 different random sites, we were able to find how many different species. Who's got a good count so far?
47.
47 different species or 47 different organisms? Or was Mr. Tout just making up numbers?
No. Anyone? How many species do we have so far?
I wrote this 34. Mackenzie says 8 so far.
Take off your virtual background. Nice. Okay. So, is he muted to you?
Oh,
Mr.
sorry,
Todd, I
I'm
think you're muted.
Thank you. My apologies. So important question. If you were to do some simple random sampling of your own, how many trials should you do if you want to collect meaningful data?
Spotlight.
At least 3.
Excellent. At least
Beautiful.
3, the magic number. All right, we're going to move on to stratified random sampling now. Are we good with that? Okay. So I'm going to just quickly show you the hillside we're on. Maybe.
You're all spotlight.
How do I had it earlier.
Miss Danita is actually taking really good video right now.
Okay, that's fine. So as we look at this hillside, right, I see sort of 3 different regions that catch my eye that appear different from one another. There's this rocky region right up and down that is likely man-made, but it is its own, it's separate, right? We have what I'm going to call my little grove, right, of these tall plants with the golden flowers that you guys found via simple random sampling with Mr. Dutton, right? And then we have almost the entire rest of the hillside, which to me just looks, it isn't a specific grove of this species, right? So I sort of see 3 different spots that I would want to do stratified. That's my stratification, is I'm picking 3 spots in which I want to randomly sample. So should we walk through at least one of those or 2 of those locations? Or?
So we're done with collecting the data that you are going to use for your lab. Now we're going to show you, so stratified random sampling is one of the other 4 techniques that we're going to show you for collecting data. So we're just going to show you how to do those. And so ask questions as we're doing them. There's a train going by, but ask questions as we're doing them so that you have a good understanding, so that when you collect data on your own, which you will be doing, that you will understand which technique to use based on the situation. So if you're collecting data in your backyard, or you're collecting data on a tree lawn, or you find some sort of field, or you find some area that you can use, a park like Edgewater, that you can use to collect data, that you understand the techniques well enough to be able to choose the one that's going to work for the area that you're collecting. So let's show them stratified random, right?
Yeah.
So
I'm actually going to go over to the rocky area. Oh. Can I go over to the rocky area?
So the Y and X has to do with the amount of space that you have and what fraction of it that you're sampling. So we'll get into that when we do the last one, and that'll help you understand how to estimate what percentage of the area that you're sampling. So for example, we just did 2 frames worth.
Mm-hmm. So we did 2 squares worth. In terms of the square footage or the square meters, whatever, of space that we measured.
That's 1 square meter. And so we sampled 2 square meters. And then if we were doing this for our internal assessment, we'd actually have to measure the entire area. We'd have to get an approximate. And so you might say that it's like 1,000 square meters. And we sampled 1 out of those 1,000. So that would be your X and Y values. But because it requires a little bit more math and doing, we're not going to get into that until the last example here. All right. So let's follow with the second technique, which is stratified random. Go ahead, Mr. Tout.
Thanks. So for instance, in terms of stratifying this hillside, remember I picked out this grove as sort of different than the rest of the hillside, right? So I'm going to still use a pretty similar procedure in terms of I'm not going to pick a specific spot within this area that I have stratified out. I'm going to still use the random sampling within it, but I'm just going to toss this in there. And now I've got to work my way to it and try to work this down to it carefully. One example of an advantage to using pencils is, right, with all of these tall plants, this is going to be a little complicated for me to do without damaging the plants, and that's important. You don't want to beat up the area that you're studying. That's one of the core theme, or one of the core messages we want to get across, is you have to be thoughtful about the way in which you collect data and the impact you're having on the environment as you do so.
And so with the stratified random sampling, what we're doing is we're comparing the different areas. So we're comparing this area that Mr. Tout is in with the rocky area, with the grassy area, and then we're going to evaluate what the biodiversity is between those 3 areas. I was seeing if I could capture a grasshopper for you guys, but he's being very elusive. All right, so then that would mean that I would go over to the rocky area next, and I would, I can toss my beanbag, I can toss my ball, whatever I have that will stay in one place. And using that beanbag, I can then sample the 1 square meter area approximately. And this rocky area is not very biodiverse. Areas don't have to be very biodiverse for you to sample them. So don't think that you can't choose a place that doesn't have a lot of plants or a lot of animals. We see here, there's probably like 4 or 5 different species in this area. It's not very many. But there's still some biodiversity, and we noticed that the plants that are growing in here are very different than the plants that are growing over where Mr. Trout is, because this is a rockier area. So I'm not actually going to follow through with collecting this data. I just want you to see this technique. And while Mr. Tout is getting some data over his way, we're going to go to the grassy area, and we're going to see how you can actually find significant biodiversity even in a lawn. All right, Mr. Tout, what do you have? You're muted.
So, first of all, do you want me to turn on my video so you don't have to work your way in here, or? Mute. Now, there's a way to rotate, right? Here we go. All right. So, what did you call the tall golden leafed thing that's not goldenrod?
That wasn't marked. Was it?
Okay.
Was it Peter? Or is that?
It was
David?
Peter.
All right, Peter.
Peter.
Yes.
Okay.
All right. So we have, and I just to get to show you that they're in different life stages here, right? This one is got the golden colored top to it, but this one is actually ready to spread, right? You see how this is set up? I'm trying to get that so it focuses. May not happen. But so this one is the same species, but it's actually ready to disperse all of its seeds when the next, heavy wind comes through. And this one looks like it already has. So they all look slightly different. I do have a couple of bugs. I don't know that I'm going to be able to find them again, but I certainly found some good bugs in here. So we definitely have some good examples of the sort of animal kingdom in terms of wildlife. I had a cricket, I had a couple of mosquitoes, and I'm of course not seeing any right now. Here's a spider. Well, there's no way the camera's going to pick that up. But we have a tiny little spider on this blade of grass. So again, lots of things here. Some other unique plants that I hadn't seen in the other area. I really love this spiny guy. Almost looks like a single branch of a fir tree. So we're going to call them piney, right? And I didn't actually finish counting them all up. I'm not that fast. So I don't have data specifically to report, but I do want to take this opportunity to clarify. There's sort of two things that we're talking about here, right? One is your IA. We're trying to give you some ideas about ways you could go collect some data for your IA, and hopefully ways that are kind of fun and you would enjoy. But then ways you would also have some significant quantitative data to talk about with your IA. Obviously, if you want to stay with doing a water study or a soil study or an atmosphere study, you can. But that being said, there is a separate component to this, which is the actual lab report for this assignment. So you do, whether or not you decide to switch your IA to a biodiversity study, completely up to you. But you still are going to have to do at least one of these techniques in order to do data collection for this lab. I don't think I'm going to be able to get a good shot of this nest, but I do have a nest.
And so to make sure that we have all of the different,
yeah, you can't see it.
you
Sorry.
could spotlight my video. To make sure that we have all of the techniques outlined. Great. Then I wanted to show you, and I'm up the hill. All right, just to show you where I am. And I already tossed the beanbag, and now I'm making my 1 meter square, which is, approximately the size of what I'm showing you right now. And then, so this is lawn, right? This is lawn like a lot of people have in their backyards, but there's a lot of biodiversity here. Among other things, we see that there's a dandelion, right? We see that there's clover. So it's this guy here. All right. And there's different grasses. we have all sorts of different grasses here. We've got the broadleaf, and we've got the medium leaf, and we've got the thin leaf grass. And so this is where estimating the numbers comes in really handy. You don't want to sit here and be like, wait, how many clovers are there? I'm being told that I cannot be heard. Can you hear me?
Yeah.
Okay, cool. So the estimate here, if there's a couple different ways you can do this, and this is the last thing that we're going to show you today, is how to actually estimate a total number for a space this big. So we'll get into how to actually do that in a few minutes. But if we look really closely down here, we can see there's an enormous density of clovers. You could easily estimate that within this 1 meter space, there's at least 1,000 of these guys. So as long as you're getting it approximately right, that you're saying that it's about 1,000 versus 100 versus 10,000, then that's fine. But don't get it off by a power of 10. That power of 10 is actually really important when we do our calculations. So if it's 1,000, that's fine. If it's 100, that's fine. Even if you're off by a couple hundred, then that's much better that you said it's 100 than if you're off by a couple thousand. So get an estimate for something that is as plentiful as grasses and clover when you have like a lawn area, because it's really, really dense. But we do have we have one dandelion plant that has a few different flowers on it. So there is a significant amount of biodiversity even in a lawn. So you can continue to do that. Over to Mr. Tout. I think you're muted.
All right, I'm going to start over since you guys weren't listening the first time. Sorry. All right, so as a quick example of another strategy that you can use, right, to do biodiversity, this is not really the greatest setup, but it can help us visualize. If I can turn around my camera, there we go. So what I have done is I have measured out 25 meters of string. Right, so I measured out the string in advance. That makes my life easier so I don't have to actually count it out while it's spread out. Literally, I just wrap the string around a meter stick and I measure it out 1 meter stick at a time, or 1 yardstick at a time if you've got it, or if you've got a ruler, do it 1 foot at a time. But if you guys can see what I have here, this bright yellow string, I hope it's showing up on the video. I've measured out 25 meters in what's called a line transect. So I've picked this spot, right? This is not stratified, what's the word I'm looking for? Systematic, right? This is a system, right? Rather than random, I've gone systematic here. And what I can do is as I come along this, right? As I come along, oh, that's better. Good call. Mr. Dutton is supplementing the yellow wire with something a little more visible. This orange tape, right? And so we have 25 meters of orange tape. So we also know, right? For instance, you could go onto Google and find out how long this trail is, right? How long is this paved trail in the metro parks? How far does it go? And in 25 meters, right? I have a very small sample size of that, but I could start to go through and figure out how many different species do I find in this 25 meters, right? And I would have to go fairly slowly and stop, look for insect life, right? Try to get an idea of what is growing along the edge, right? And right now I'm mostly seeing just different types of grass, or really just two types of grass. But the point is, this is a line, and I'm really looking somewhat one-dimensionally, right? In terms of just, I'm not going out very far on either side of the line, maybe a foot or two. Whereas if you look at the whole sidewalk, obviously that's not your goal, is to do a biodiversity study of a sidewalk, but this is more of a belt transect. So if we had put the line right down the middle, instead of having a line transect, we're really only looking a few inches to each side. You can do what's called a belt transect, where again, you want to pick a relatively short distance, like say 10 meters or something, right? Or 30 feet. And then maybe do that at 3 different places along a space. Or if you can imagine this orange tape just going right up the hill, right? If we had just taken the tape, oh, go ahead, Mr. Dutton.
Sorry about that. So something that we should probably do is actually we're going to put this line in the woods, and then what we're going to do is we're going to measure all of the trees that are within 1 pace of the line, and that's going to help us count the number of trees approximately that are in the wooded area in that direction. So I'm going to pick up this line. I'm going to pick up the orange tape real quick. And the videographers here will join me in the woods super quick so we can do this line transect.
And again, to clarify, guys, looking across the beach, I can count the trees, right? Oh, that's not so bad. I can do that study. And of course you could. Now, looking at the forest, who wants to take the time to go through and try to count all of the trees? And even if you did decide you wanted to spend a week of your life doing that, how would you know you didn't miss one, right? How would you know how accurate you really are? So the reality is you can't be, right? You can only be so accurate. And that's why we use math. So as opposed to going through this forest and trying to get a count of every single tree that's in this forest, do you want me to hold one end? Oh, you've got it all.
Nope.
He's literally going to just put a line through the woods. And then he's going to, like he said, take one big step in a direction. Or as he goes down the line into the woods, he can just estimate how many trees you have in one section. Here you go. I got you. So I don't know how clearly you can see Mr. Dutton disappearing into the darkness. There he goes. I hope he holds on to this rope or he's never coming back.
So it's a little tricky to make this line straight, right? Because there's lots of plants in here. There's lots of trees, lots of shrubs, but it doesn't really matter if it's perfectly straight. Mr. Todd is helping me make
If you
sure
want to
that
call it that.
I'll turn on my video here in a second so you can see it from my point of view. But then what I'll be doing, as I said, is I'll be counting the number of trees that are within 1 pace of this line. And that will help me estimate the total number of trees that are in this forested area. Mr. Tao, it's okay. You can just cut it right there. You can just split it in the interest of time. All right. So, hey, there's Miss Danita. So here is this line that is going through the trees, that is not going through at all straight, but that's okay. So now I'm going to find all the trees that are within 1 pace of this line as I travel the line. This is called a line transect. It's up to you to determine the distance that you want from the line. I said 1 pace because I don't have a meter stick with me, because I don't have a measuring tape. You don't have to have these in order to be able to do this kind of sampling. Just, you have to be consistent, and that's what it comes down to. So this is 1 tree, clearly. We've got one right here. And within 1 pace, I definitely have another right here. I've got 2 more right here within 1 pace. I think this counts. So far we're up to what, 5? And then within 1 pace, let's count it, 6. And here we have 7 and 8. And I just walk along the line, and anything that's close, I can go ahead and I can see if I should count it. So that's 9. And then here's 10. And let me make sure I got the line in view. 11 and 12, 13. This one is within 1 pace. 14, 15, and 16. We've got 17, and I'm going to accelerate a little bit. 18. It's kind of hard to ignore that one. And then this guy is within 1 pace, so that's 19. Am I spotlighted right now? I don't think I am. Okay. And then we've got 20, and I'm sure you can see a variety of video from Mr. Tout as well. Uh-oh, headphones. And then as I go up, I notice that there are no trees in this particular area. That's a branch, that doesn't count. And these are too far away, that's more than a pace. But here is 20 and 21. Those are not trees, I'm only counting trees. This counts, this 22. And then I've got 1 more over here, that's within 1 pace, so that's 23. So along my line, I have 23 trees. And if the line were 20 meters long, and I could estimate that distance, if the line were 20 meters long, and I was measuring 3 meters on each side, then I can use those numbers to calculate about how many trees there are in this entire area. And that's where the X and the Y come into play. That's where those measurements come in. And so this is an example of a line transect. Belt transect works pretty much the same way, except instead of just pacing things off from the line, then what you're doing in a belt transect is you're actually running 2 lines that are parallel with each other. So we might run another line like over here, and you would measure everything that's in between those 2 lines, and that would be a belt. So you just find everything that's within that belt. As you might imagine, that could take a while. But if you were looking for a specific species, that might not take as long.
Brian muted. All right, there we are. If you're interested in trying to do animal studies, the belt transect is a little bit better because, again, you're looking for a specific species. You're not going to try to count every bug or every plant in the forest, but trying to find rabbit holes or squirrel nests up in the woods can actually be a little bit harder to find, you might imagine. And so sort of defining a belt that you're going to walk and just look for, like a specific species of mammals, it works pretty well. If you're trying to just figure out how many species are there total, belt transect is like Mr. Dutton said, very time consuming. So I did find an interesting species. What did you say, Ms. Danita? Monkey ball? Yeah, that's all I know either. It's the only, so I am curious if anyone can figure out what this is for me. We found one of these along the line transect. And I did not try to identify where it came from in terms of which tree it fell from, but it is a pretty cool looking species. While Mr. Dutton is setting something up, do you guys have any questions? Anyone feeling adventurous in terms of exploring biodiversity with their IA now?
Does it just seem like a lot of work?
It always seems that way. But the data collection piece is the most fun of all of these. I know it's hard to imagine having fun at school. As soon as you have to do it, it's no more fun. But I'm having fun right now.
I don't have audio on, do I?
Yeah, you're on.
Oh, sweet. So what I'm doing right now is I am making a grid for the quadrat. So I'm trying to just demonstrate how it is that you can make this grid. Oh, it's a brain. And this is not perfect, but the idea being that if we separate our frame, which again, if you want to borrow the frame, we have several that you can borrow. Just let us know. And I just made the frame in one dimension, but if I had more time, I could make the frame going this way as well, and it separates it into squares. So now what I can do with this frame is if I end up in a spot like this lawn that has, you got this video? Spotlighted? Okay. If you're in a spot that has a lot of biodiversity, like this lawn area, this grassy area, then what you can do is you can section it off and then measure random sections. There's a couple different ways. You'll see at the end of this document that it separates into 25 sections, and every section corresponds with a letter. And so if I were going to use like my name, I might find section J, section O, section H, section N, and section D. And those would be the 5 spots out of the 25 total that I would sample. Alternatively, you can use like our beanbag or, the ball or whatever. You can toss it wherever it ends up. That's where you sample. And we use a little brain too. And so, for example, we could toss it 5 times. We could sample all 5 different places. And there's a total of 25 squares. Then we just sampled 5 out of 25, or 1 out of every 5 squares. That's 20%. Because it's 20%, or because it's 1 out of 5, we can multiply our results by 5 to get the total amount. So that's what I was saying before about, oh man, counting all those clovers is really going to take forever. We don't want to have to do all that. So we can just take 3, 4, or 5 of these little areas, and we can go, all right, I look down here and I find that, here's a plant. And when you get down to the roots, you see that there's not as many actual plants, that the blades of grass don't correspond, 1-to-1 with plants. And we find that in this area, there's 40 plants. Well, and if we find there's 40 over here as well, and we find there's 40, we have an average of 40, and we multiply that by 5, and we can then say that, or if we get 40, 40, 40, 40, 40, that gets me, 200, and multiply that by 5 for the whole thing, and that tells me that there's 1,000 in here, all right?
She says she's going to stick with water then.
To be clear, we can help you a little bit with understanding the math.
Yeah.
Sorry, I was.
There's a lot that we can help you with the math, but this is really a helpful technique if you have a high number of species, and you really need to get a semi-accurate count. Any questions about the quadrat sampling technique?
No.
So let's
So let's reinforce real quick. Let's just conclude, right?
This
This today
serves
serves two purposes. The first purpose is to make sure that you can do this lab that we're starting today, and that is going to take like 2 to 3 weeks to finish. This is the first site. So at the beginning, when we did all the random sampling, that's the data that you're going to use. And then the second purpose is the internal assessment.
So sorry. Yeah, for the internal assessment, you want to think about what you're interested in and what you're curious about. There are advantages and disadvantages to every type of testing, right? The baseline, the bottom line is you have to collect some data, right? Again, going out and collecting water from 5 different places across the city is one way to go about this, and then doing tests at home on that water, right? Now you're dividing the tests up into several steps and locations. The advantage, of course, being that the amount of work you have to do outside of your house is limited, right? So for some of us, we really don't want to be out of our house very much right now. For others, I feel like I'm kind of itching to get out, right? What can I do that would be a good excuse to go outside for a few hours and walk around the park? So this would be, an excuse to go, you know what, I'm going to go do my data collection. And then the analysis I have to do at home is just math. And if I'm not sure on how to do the math, I can pester Mr. Dutton or Mr. Tout with a little bit of help just in terms of like, what are the calculations supposed to look like? I did some data collection. What does it all mean? And that's stuff that, through doing this lab in small groups, you'll have a better idea of, and you'll have some opportunities to pick our brains through doing this lab. And then you can apply what you learn to potentially your IA if you so choose.
So hopefully that gives you at least a good introduction. Of course, we will explain more in class. And so any questions that you have, I know it's a lot of information and it can be overwhelming to try to make sense out of all of it at first. It really helps if you can try a little bit of it on your own, which is why, the lab that we're asking you to do, you are going to be trying at least one of these sampling techniques. So please let us know if you have any questions. Don't be afraid to email us or to put any comments on the Google Classroom. And we will see you either tomorrow or Friday. Oh, he's totally giving me bunny ears behind me. That's hilarious. Hold on.
I think I'm in the sun.
All right. But yeah, if you have any questions, unmute. Otherwise, have a wonderful day. And
Have a wonderful day.
You too. It's so sunny. At least
Have a good
hopefully
day,
it's
guys.
getting sunny where you are.
You guys are rock stars.
Bye.
Bye.